our group has created an efficient, portable, real-time, and minimally invasive way to perform blood testing without the need for a hospital lab. We are developing a wearable patch that can measure many of the same biomarkers that we would use in an emergency department, starting with the cardiac enzyme troponin. And troponin testing is currently done by drawing blood at certain intervals over several hours. We can measure this continuously through the wearable patch. And this technology could change pre-hospital care, remote care, and space healthcare, along with tertiary care, by reducing the reliance on a lab to provide this critical information.
We use the standard electrode composed of the working, reference, and counter electrode integrated onto a single chip. We are able to detect the troponin level by functionalizing the gold working electrode with the troponin antibody and glucose aptamer. This aptamer is tagged with a methylene blue molecule in a way that upon binding with glucose, the methylene blue will move closer to the gold surface and generate an electrochemical signal. In our first condition, when troponin level is high in the interstitial fluid, glucose is obstructed by the troponin, so no glucose can reach to the aptamer and no signal is generated. With a low level of troponin that is still higher than the normal range, some glucose can reach the aptamer with less troponin blocking its path. This leads to the detectable signal. Lastly, with only glucose present in the interstitial fluid, all glucose molecules will bind to the aptamer and this creates the highest signal. Now we can return back to the lab for the demonstration. As we explained using the schematics, we functionalized all the chips with troponin antibody and glucose aptamer. Then we spiked different troponin concentration into the target solution to have high level of troponin, low level of troponin, and no troponin. Now we drop deposit the different targets on different electrodes. After incubating the electrodes with different targets for 15 minutes, we can measure the signal generated by glucose aptamer. As you can see, by decreasing the troponin concentration, the magnitude of the signal increases.
Here is our microneedle patch. On the heart tracker, it will be applied on the skin as a pinless electrodes for extracting and sensing skin interstitial fluid. Our microneedle is a 7 x 7 mm square and consists of 100 pyramidal needles. The needle length is only 0.8 mm. It won't cause any uncomfortable feelings or bleeding after long time skin penetration. The microneedle patches are fabricated using PEDOT-PSS hydrogel. It is a well-known conductive polymer and showed great biocompatibility in our cell and animal tests. So our microneedle is safe for long time skin application. Now we are showing the microneedle fabrication process. This is our silicone molds. The mixture of PEDOT-PSS and gold nanoparticle is added on the mold and evenly distributed on the silicone surface. Next, a copper wire was applied on the microneedle. Then the microneedle is dried under room temperature. Fabricated microneedle patches can be stored in a vacuum-sealed chamber under room temperature. The sealing process is very easy and quick. No special storage condition is required. Before moving to the sensing experiment, we tested the robustness of our microneedle patch to ensure it can undergo rough handling or transport conditions. Here we utilize agarose phantom gel to mimic the human skin. Microneedle can be inserted into the agarose with a finger pinch force. This process can be done by one hand. From the back side of the agarose, we can see the needle penetration trace. Our basic sensing system consists of 3 electrodes. The PEDOT microneedle serves as a working electrode, a silver microneedle is used as a reference, and a gold microneedle works as a counter electrode. The 3 electrode sensing system is connected to a potentiostats that generate detectable redox signal in response to the target concentration.
Having access to a point-of-care test like this patch in remote locations of Canada, where we oftentimes either don't have access to a lab or have significant staffing shortages, could help us identify medical emergencies quicker to treat the patient and medevac them to a tertiary care center faster. And because this technology is so light, small, inexpensive, and easy to apply, it has great applicability to both healthcare in space, as well as rural and remote locations in Canada.